Today I'd like to show you some recent data from my lab and basically highlight one point that is keeping us up at night. That's something that systems neurobiologists will have to deal with. We will have to deal with over the next 10 years or so. And the people who've collected most of the data that I'll show you, shown right here, that's Dan O'Connor, Daniel Huber, and Simon Perron, the first two actually on the job market. So one important theme in systems neurobiology is to relate patterns and define action potential patterns and defined neuronal populations to specific aspects of behavior or perception, so-called coding problem. And that's often done by correlating different aspects of these trains of action potentials, the number or the timing of the spikes to behavior or in various kinds of experiments. But I think it's pretty clear that that's not a terribly well-constrained problem, that we need to go a step further and to causally relate action potentials to behavior. We need to take control of the firing of these spikes and manipulate them in the context of quantitative behavior. And there's a lovely tradition in the awake behaving primate field of doing just such experiments. And perhaps the best example is illustrated here. This is from Rodolfo Romo's lab, where a monkey sits in a chair and he has to do a so-called flutter discrimination task. So stimuli given to the digits, and it's a working memory task. The monkey has to distinguish whether or not the first or the second of a stimulus is higher in frequency. Can learn this very accurately. And then at the same time, Romo's lab records from somatosensory neurons, so-called in the somatosensory cortex, so-called rapidly adapting neurons that are hypothesized to encode the flutter frequency delivered to the digit. When they stimulate in the vicinity of these neurons with an extracellular stimulating electrode, they can replace one of the stimuli with an extracellular stimulus, and at the level of behavior, the monkey doesn't miss a beat. And this is a remarkable experiment, really causally linking action potential patterns in specific cell types to behavior. It's an amazing experiment, and this kind of clear example is also very rare, and that perhaps shouldn't surprise us, because with an extracellular stimulating electrode, you excite an entire column. This is now a column, a wiring diagram, a circuit diagram of the major excitatory connection in somatosensory cortex from the mouse. When one does these kinds of experiments, one non-discriminately stimulates all of these cells, which are connected in highly specific circuits in an unnatural way. In addition, of course, GABAergic cells, which are not shown here, and fibers from many sorts of places. So the stimulus is not even spatially localized. And so in the mouse, we can put together these kinds of wiring diagrams made up of specific neuronal population. And one of the great advantages of the mouse, and in part due to the wonderful work ongoing at the Allen Brain Institute, we have actually mice with that express Cre in specific neural populations which correspond to these nodes in these circuit diagrams. And these mice are very useful to us because they allow us to express switches and sensors in specific neuronal population, and perhaps one-up the kind of experiments that you just saw, and really do circuit analysis at the level of defined cell types, examples of which you've already seen during this talk. I should mention that in this kind of wiring diagram, even in one cortical column, even though there are individual cells here depicted in this diagram, of course, this really corresponds to and really represents statistical connectivity corresponding to groups of neurons sort of on the order of 100 to 2,000 neurons. There's about 10,000 neurons in a barrel column that is in the one cortical column in the mouse somatosensory cortex. So with these kinds of tools that give us access, genetic access and functional access to specific cell types, we and many other people in this room and across the world are now doing this kind of causal circuit cracking, where we try to record from specific cell types in the context of a quantitative behavior. And that might give us hypotheses about what these neurons might encode in relation to that behavior. And of course, to test these hypotheses, we have to manipulate activity, we have to turn activity off, see if that perturbation perturbs the behavior. And of course, perhaps even more informative, we'd like to drive activity in various directions and thereby manipulate behavior in a way consistent or inconsistent with the hypotheses that we set up. Now, today, and we're taking this research program very seriously. I won't have much more to say about this, but I think if we imagine that just mapping genetic and developmental cell types, that we've somehow captured the diversity of response types and complexity of the brain, I think we're kidding ourselves. And of course, there are many examples of that in the literature. But wherever we look, even at defined neuronal populations in one particular region, we find that multiple representations, sparse representations, are often intermingled within the same cell types and same parts of the tissue. And dealing with this problem is something that we'll have to do over the next 10 years or so. And I'll give you a couple of examples of that based on imaging experiments, cellular level imaging experiments derived from mice doing an object localization task. I have to tell you a little bit about the behavior. So mice are head fixed, and the head fixation provides behavioral control, but also allows us to use recording techniques that have less biases than standard extracellular recordings, namely cellular imaging, where you just can look at basically every cell, and patch clamp recordings, where you hunt for cells based on them just being in the vicinity of the electrodes rather than based on spike patterns. It's a head-fixed behavior where the mouse has to use its whiskers to measure the distance to an object coming within reach of the whiskers on one side. And there is a target location in response to which mice have to lick for water reward, and a distractor location in response to which they have to hold their tongue. And mice learn this relatively quickly after a couple of days. Here's an example of a couple of trials, and you'll see in the first example, this is a no-go trial, a no-lick trial. The animal correctly holds his tongue because the pole comes within reach in the distractor location. In the second trial, it's a hit trial, and you saw that the mouse correctly licked for a water reward. So since this is a head-fixed behavior, so animals learn this over about a simple version of the task in the first day and become experts after about 7 days. And there's a few things I'd like to tell you about this behavior. It's dependent on the whiskers. It's all done in the dark. When you cut the whiskers, behavior goes to chance levels. If you lesion reversibly the somatosensory cortex or the motor cortex, where the imaging experiments are done that I'm about to show you, performance also drops to chance level. Now, here is how an imaging experiment is done. So basically, the behavioral apparatus is simply moved under a 2-photon microscope, and you've seen examples of that, similar examples for the fly, sort of an emerging paradigm. And here we use a genetically encoded indicator of activity that is based on calcium imaging. So this is a protein probe that is introduced with viral reagents into a big chunk of the brain here, and we can image individual cells. And when cells fire action potential, calcium comes in through the membrane, and we can pick up fluorescence changes of the protein sensor as an indication of the activity of the cell. So in the movie that I'll show you, a bunch of trials were concatenated one after the other, and when the pole is present for exploration, you'll see a white square flash in that movie. It's sped up several-fold, and you can already see that in a tri-locked manner, a bunch of neurons come on and flicker on and off. And this is essentially raw data. The only thing that we've done here is some movement control. Remember that this is an animal doing things, responding to a stimulus. And so the movement correction introduces these black lines that you saw fly by. Now we can now extract the fluorescence transients corresponding to activity in individual cells by drawing regions of interest around individual cells. And we get traces like these that already illustrate this heterogeneity that I'm talking about. The vast majority of these cells are layer 2-3 pyramidal cells with, as far as we know, identical, more or less identical gene expression patterns, similar developmental history. And here's a cell that fires robust calcium transients in a task-related manner in a neighboring cell that is essentially silent, in a couple of examples below. And I should say that with these kinds of measurements, we see robust, reliable fluorescence transients to bursts of action potentials of 3 or more action potentials within a window of about half a second. These kinds of imaging experiments have some important advantages compared to electrophysiological measurements in the sense that we can image densely and really sample cells in the vicinity in a field of view. And we also know the spatial relationships between neurons. And spatial relationships, because we know about the spatial connectivity patterns in the cortex from in vitro recordings, spatial relationships can give us hints about the circuit mechanism underlying these kinds of response types that we see. And of course, since individual cells have addresses in terms of location, we can come back day after day and reliably record from the same neuron over timescales of learning. And in these kinds of fluorescence imaging experiments, we can combine them with markers of cell types in transgenic animals or do post hoc analyses to relate firing patterns to neuronal cell type. Now, I should say, of course, there are disadvantages to these kinds of techniques, and the main disadvantages are that the signal-to-noise ratio is still inferior to electrophysiology. We don't detect the lowest firing rates. We can't detect individual action potentials. It is difficult in these kinds of bursts to distinguish the bursts that are produced by 3 action potentials, say, from the bursts that are produced by 4 action potentials. So there's a limited dynamic range to these kinds of measurements. So it's really an orthogonal technique to extracellular physiology. So here are some of the brain regions underlying the object localization task. Here's the famous barrel cortex that you've already heard a lot about. And this is strongly interconnected with the bristle motor cortex that makes the whiskers move. And the connections between these cortices happen at the level of layer 2-3 pyramidal cells. And those are the cells that we'll try to image. And we think there might be the prime sites for sensory motor integration, because this is where sort of the place that is involved in the control of voluntary whisking, of control of the location of the whiskers, and where sensory input converge. So now let's look at layer 2-3 pyramidal cells in awake behaving mice doing the object localization task. This is now an expert mouse that knows how to do this task very well. It's been trained for 10 days. He works at 95%. And here I've plotted the neurons based on how selective their activity pattern is based on object location, based whether or not the object, the pole, is in the target location or the distractor location. And you can see that there are a bunch of cells here. 3 are highlighted here that are highly discriminative, that even on individual trials could tell an ideal observer whether or not the object is in the go location, blue traces here, or in the no-go location. Highly discriminative neurons, but there are many other neurons that don't seem to care about object location at all, and most of these neurons, in fact, fire very few or no action potentials, even in a awake behaving animal doing a somatosensory task. So you might think, well, the majority of cells are here pyramidal cells, about 85% or so. Could these discriminating neurons correspond to some rare subtype of GABAergic cells. So what we can do after one of these imaging experiments, use histochemical markers of GABAergic cells. Here's one example. This is simply GABA staining. And just to highlight that pretty much all of the cells that discriminate object location actually belong excitatory pyramidal cells. So there's a sparse subpopulation of pyramidal cells that code for object location in layer 2-3 of the barrel cortex. Now you might say, well, these are relatively poorly vetted techniques. We might miss some stuff with our action potential, with our fluorescence sensors, and that is indeed the case, but qualitatively the picture is identical if we probe cells in an unbiased manner with patch clamp electrodes. And here is about 3 dozen or so layer 2-3 neurons that were recorded with loose seal cell attached recording. And here we rank order these cells based on how well these neurons, now based on spike trains recorded with bona fide extracellular methods, how well the spike trains can distinguish object location on a trial-by-trial basis. And we find, again, that a small fraction of neurons is highly informative about object location. Many neurons don't seem to care at all. And in fact, not surprisingly, there's a very good correlation between the neurons that are informative about object location and the neurons that fire appreciable spike rates. So again, there are lots of neurons that don't fire basically sufficient spikes to be informative about object location. We've done the same kind of experiments during learning, where we start with completely naive animal. I don't have time to show you this, and look what happens over learning. And the hypothesis was that perhaps the sparseness in this layer 2-3 cell network really is sparsened with learning. It's produced with learning, but that's not what we find. We find that there's a sparse subset of neurons in naive animals and in expert animals that are sensitive in the somatosensory cortex to deflections, to forces, temporal forces exerted on the whiskers by the pole. And what animals do during learning is they adapt a more and more intelligent whisking strategy and interact with the pole in novel ways, in such a way that contact forces, differences in contact forces that are location dependent, are translated into changes in activity, distance or object location dependent differences in activity in layer 2-3. And it is through this contact force responsivity of these neurons that these neurons, in fact, code for object location in the primary somatosensory cortex. Now, again, it's few percent to 10% of neurons that show coding of object location. So one question one can ask is whether or not this very sparse activity patterns in these layer 2-3 cells can be read out by the animal. And an experiment that we did now a long time ago really suggests that animals can detect and perceive ultra-sparse activity in layer 2-3 cells. So the experiment that we did here is we labeled a sparse subset of layer 2-3 pyramidal cells with channelrhodopsin, mimicking the activation pattern that we see during somatosensory behavior. And we find that it takes about 1,000 action potentials to drive perception of these optical stimulus now to about 80%. And that corresponds very nicely to the modulation, to the extra action potentials available, or the difference in the number of action potentials across the entire layer 2-3 network during the object localization task. Mind you that we have not been able yet to stimulate using channelrhodopsin the same neurons that are activated during the object localization task, and that's a major goal for the future, and will, I think, be required to causally relate ultimately these layer 2-3 pyramidal cells to somatosensation in these sparse networks. Let me show you another example from the primary motor cortex, which in some sense makes this point even of sparse coding even more strongly. Here, what I'll show you is 3 examples of 3 units that are representative of intermingled sparse networks in layer 2-3 of the motor cortex. I should say this is rather preliminary, but I think the basic message will hold. So here's the learning curve. This animal, we start with naive animals. Here is his whisker pattern. So in blue is on one trial type, where he basically whisks after the pole accidentally hits the whiskers on correct go trials. And then in an expert animal, he whisks when the pole comes within reach on both trial types. And there are units in the motor cortex, not surprisingly, that really seem to drive whisking, okay, that correlate with whisking, and the activity pattern shifts earlier during the period of when the pole is present, when exploratory whisking occurs. There's another bout of whisking which we don't think has to do with cortical activity, which happens during consummation of the water reward. So there are whisking-related units that seem to correlate and perhaps drive whisking in the motor cortex, not surprisingly. But there are other intermingled units, and that's only about 10%, the previous portion. Here is a neuron that is active during early. The animal doesn't know about the task. Sometimes he gets the reward, sometimes he doesn't. On the blue trials, he gets the reward, and these neurons seem to be active after consumption of the reward. This is the answer lake after which the reward is available. But these units tend to fire on correct trials and seem to shift forward with learning until in an expert animal they seem to anticipate and perhaps drive the licking, which is the behavior, of course, to get the water reward. And here's a third kind of unit, again, on the order of 10% in this complex network, that seems to be activated by contact, but only in naive animals. Remember that the motor cortex receives strong input from the sensory cortex, and we think this really looks like an error signal to us. Unexpected contact produces responses, but when the animal starts to know the task and expects contact with the pole during the exploration period, this activity invariably goes away, the contact-related activity in the motor cortex. So we're excited by seeing all these activity patterns because there's been a lot of work in the theory of motor control, bolstered really by lots of psychophysical experiments, that the brain must contain an inverse model, essentially, of the mechanics that allows the brain to drive actions to get a reward. But for stable movement control, the brain also needs a forward model of what the sensory consequences of that movement might be. And the kinds of activity patterns that we see here intermingled in layer 2-3 of the motor cortex have signatures of both these inverse and forward models of motor control. So these whisking cells, which change their patterns to drive more intelligent whisking in an expert animal, it's an example of an inverse model. Similarly, reward cells that then seem to drive licking for the water reward, again, an example of an inverse model. And these contact force cells, which happen only when the contact is unexpected and then go silent, could be an error signal helping to tune up the forward model of motor control. And I should emphasize again that these representations are sparse, all involving roughly 10% of the neurons. They're intermingled, so neighboring cells of the same type can have different response types. And we also have evidence that as the animal learns, the responses become more reliable, and there are, based on the statistical nature of the activity, that these are actually Hebbian networks, that Hebbian plasticity helps to tune up this kind of learning-related plasticity and strengthens connections between neurons of similar response types, but not connections between neurons of different response types. How do we move this kind of activity pattern to causal neuroscience. We basically, everything that I've shown you are correlations, and systems neuroscience is full of these kinds of studies. The approach that we're taking, I would call, is a brute force approach. We think we can actually map in this system all of the cells that respond and play a role, at least in the superficial layers, that we think play a role in this behavior. And then because we know their addresses, the spatial location, we can target them for optogenetic manipulation, or perhaps even more simply for ablation, and look what happens to the network in response, what happens to behavior as we ablate an increasing fraction of neurons that we think represent a particular aspect of the behavior. We think that this is going to be very exciting over the next years, but it's quite limited because, as many of you know, these kinds of cellular imaging experiments are still very much limited to the superficial 600 microns of the motor of the neocortex, only about 10% of the mouse brain. And here is my wish list. And I think what we need to do and where we need to go is develop molecular tools that allow us to access cells based on their activity pattern and based on a trigger provided by the experimenter. Light and calcium, essentially coincidence detectors, for example, of light and calcium, to mark cells that are active during specific phases of behavior with millisecond precision, because cortical computations happen over timescales of milliseconds or tens of milliseconds. But we have to go beyond marking cells. We have to actually get access to these cells and couple photo and calcium-dependent switching, or small molecule and calcium-dependent switching, to transcriptional mechanisms that allow us to then get access to cells that were active during specific phases of behavior for these kinds of manipulations that could test causal relationships in these networks that underlie complex behaviors. And I think this is a kind of program that could really be achieved. One can put together these kinds of protein systems. It's an exercise in protein engineering. On the back of an envelope, there are ansatzes in the literature, and one could really, with sort of 1,000-person years, really make a lot of headway in this kind of area. And I think it would really help address and answer important questions in systems neuroscience for many, many years to come. Thanks.